Okay, so our next notes are on cell boundaries. Like we had talked with cells and living things, one of the important organelles for a cell is the cell membrane. And the cell membrane is just the outer layer of the cell. It regulates what goes in and out of the cell, what can be allowed in, what can be allowed to leave, and sometimes this takes energy, sometimes it does not, and we're going to kind of talk about that today. So make sure you're filling in your guided note page. Make sure you've printed that out. Also, you want to make sure that you're taking notes on this as well, because I will be collecting your notebooks at the end of each semester, or when you're done halfway through the course. You need to show me your notebook. I'll give you a grade on it. But make sure you're taking good notes. Don't wait until the end. Sometimes I'm that way. I put stuff off, and in the end, I don't do a very good job. So I encourage you to take pride in this, to take good notes, so that when you're completing your readings and doing different activities for this course, that you have good resource to look back on. Okay, so again, talking about the cell membrane. Okay, when we talk about the cell membrane, there are some basic parts to the cell membrane, and they a lot of times they talk about the fluid mosaic model. Okay, now a model is just it's kind of like something that's similar. Okay, so you make a model car. It's not the car. It's similar. It's smaller and so on, but it's just a model. It's similar to it. Mosaic, if you know what a mosaic is, it's we have a teacher here that makes mosaics out of glass, and you put little pieces together and you glue them all together and you make this artwork. That refers to the fact that the cell membrane is like that. It has many different parts that work together. Fluid is that it changes. So if we were going to add these words, we probably want to do that. The fluid idea is that it changes. So based on the needs of the cell membrane changes. So if it needs to let something in, it can let it in. It changes its shape. The mosaic part refers to many different parts. Okay. And then the model is just, something that is similar. So to understand this, it tells us that the cell membrane is changing. It adapts and changes as needed, and that it's made of many different parts. Okay, and this is a model. When you look at this, we're saying that this idea is a basic description of it. Okay, so when you see that fluid mosaic model, that's what it means. Now, you have something called a lipid bilayer. Okay, we're going to learn a little bit later in the course about the different kinds of molecules that make up all living things. And a lipid is one of them. A lipid is kind of like an oil or a fat, okay, which is a basic type of organism made up of specific compounds. Okay, we're not going to worry about that right now, but it's a bilayer, which means two layers. And it has something called a polar head. So I'm going to draw you a picture here so you can see what it kind of looks like. So it has this head, and it has a little tail like that. Okay, so it has, and this is polar, which means it's charged. So it's positive on one end and negative on the other. Okay, this is kind of simplified, but I think this will give you a good idea. So then, and the cell membrane is kind of made up of a bunch of these next to each other. And I encourage you to look in your book at this to see exactly what this looks like. It may be a better picture, but I'm giving you a decent drawing here, I think. just draw this. Okay, so this is your lipid bilayer. The bilayer are two, and this just goes on forever all the way around the cell. Okay, and then there are things found within this lipid bilayer. Okay, and within this lipid bilayer, you can have things such as a protein channel or pump, and you can also have on the lipid bilayer, you can have carbohydrates. Okay, so if I was going to add this, let me just add these two things in here so you can see them, what they kind of look like. We're trying to move this. Hope it moves. It will. Okay. So then we keep going here with this. We're going to put in a protein channel, which kind of looks like this. Let's do a different color actually. We'll put a really different color. So we have this green. Kind of looks like this. Kind of look like a little Brazil nut or something, or a pecan. And then, but it's a channel. Okay. And then next to that would be more of these, this lipid bilayer. This is my crude attempt at drawing a cell membranes. Please forgive me if it's not perfect. Okay, so we have this protein channel. Let's actually fill that in so it's maybe make more sense. Okay, and we'll erase a little bit here. Okay, a lot of times these, yeah, it's too big. We'll just go like that. Okay, so we have this protein channel, and it could also be a pump as well. And then, and this just goes on all the way around the cell. And there might be different channels and proteins. It just makes a complete cell. And then all the inside here, you have all the organelles. Okay. And then so you have a protein pumper channel. And then we put a few more of these little bilayers in here. The lipid bilayer. Okay. Then on top of these, you have these carbohydrates. And they kind of look like right here on the, let's undo that, do a different color. Let's go purple. So on top you have these little, we're going to learn about carbohydrate chains. They might be all on the outside here, going in different directions. Okay, all on the outside of the cell. This helps the cell know what's next to it and what's going on next to it. Okay, and and also what you can have is on the out of this cell membrane, if you're a plant, you have a cell wall. And it's kind of a very rigid structure. We're going to get real thick here. Okay, it might be thicker than that. It's all around the outside of the, okay, let's not do the last one. Okay, all around the outside of the cell membrane. And it gives the plant a rigidness so that it can stand up and collect sun and do photosynthesis like it needs to and offer structure and support. But really we look at this, if we go back and take that out of there, that cell wall, we have these three basic parts. We have the lipid bilayer, these red parts. We can actually put that in there. Yep, not good. Yeah, I gotta go backwards though. Get rid of that. This is your lipid bilayer. This is this whole thing right here. Okay. Then you have, this is your pumper channel. Okay. And then here you have this receptor. It's kind of like tells the cell what's going on next to it. And those are all along the outside. They're made up of carbohydrates. And that's your fluid mosaic model. Each one of these parts works together to allow the cell membrane to complete its functions. So if we continue, and like I said, this just controls movement in and out of the cell. And because this is, positive and negative, we're going to learn more about that later. It allows some things in, but doesn't allow other things. Because, if they're both positively charged or negatively charged, they're going to be repelling each other, and they're going to have a hard time getting in and out of the cell. Now, any kind of movement in and out of the cell, we have some basic ways that's accomplished. One of them is diffusion. And diffusion is the movement of particles from a high concentration to low. So if we add that, concentration. It's always going to be that way. Wherever there's a whole bunch of something in the cell and there's a little something on the other side, the high is going to want to go to the low. Now, we talked back here about pumps. Now, a pump is for when, say you have a low concentration on one side and a high on the other, and you want to go from low to high. Well, you're going to have to use a pump to do that, because naturally it's not going to want to happen. It's going to want to go the other way. So they use pumps to do that. You can also see, I want to add as well, this channel. And this channel kind of has like a shape to it right here. Like if we were, and like a molecule might fit right in there. Let's make it this pink one. Okay. And if it fits right into that, it kind of changes its shape and allows this to go right into the cell. But diffusion, we're going to talk about, is any particle going from high to low. And you've seen this in your class or in your life in general, diffusion. It's like, boys trying to impress the girls. So after phys ed or whatever, they put a whole bunch of cologne on. And then they go from phys ed class, they go to social studies. And you might be a girl or somebody sitting in the back of the class, and you see Tommy come in, or whoever, and all of a sudden you get this really strong whiff of, whatever cologne they're wearing, Axe or whatever it is, and they're still across the room. That's diffusion. There's a whole bunch of perfume or cologne particles around them, but they diffuse naturally, and eventually you smell them. Okay, that's diffusion. Let's go to our next page here. Let's clean that one off. Let's go to our next. Okay. Particles high to low. Let's put diffusion in there so we don't forget. Okay. And then we have the idea of osmosis. And osmosis is very, very similar to diffusion. And but it's just water. It's just H2O. H2O only. Okay. Like you go to the amusement park, and when you were a little kid, they had these signs like, you have to be as tall as this character. Like at Disney World, I think it's Goofy. You have to be as tall as Goofy. Well, for osmosis, you got to be H2O, or you're not getting on the ride. You're not going to be allowed to pass through, or we're just talking about water. Okay, so osmosis is just water, and it's the same though. It's the same as diffusion, in that a high concentration of water always goes from high concentration to a low. The thing that's important with osmosis is to remember it's talking about the water. So if we have, for example, we have a beaker full of water. Okay. Yep, let's go back. This is H2O. It's 100% water. And then we take a baggie and drop it in this water. And inside that baggie, it's 70% H2O. What's going to happen is, because of osmosis, and as long as the baggie allows the water to do this, some of this water is going to go into that baggie because it wants to have the same concentration. Because the outside of the bag is 100%. The inside is 70. This is the lower concentration, so the water goes in. Okay? And if the water can pass through the openings in the baggie, it will. Okay? And if there's 70% water in here, there's 30% salt, whatever it is, salt, whatever. And usually what happens if the salt can't come out, this will expand and get larger. Like, it will blow up. And we're going to do an experiment, we'll see that, where the thing expands and gets bigger. Okay? The same thing if it was the other way around. So if we change this, we made a different beaker in a baggie, and we had water that was 70%, and we put a baggie in here that was 100%, the water would go out. And if the holes weren't big enough to let whatever, or I guess it wasn't big enough to let the salt in the water into the baggie will shrink. Okay? And that's osmosis. And diffusion enjoy the same thing. If you have particles, an example, we're just going to draw a basic picture here. We'll go gray. I'm going to call this a cell membrane. I'm going to get a little darker. Yeah, one more time. It's too big. I'll get this right eventually. Too big still. I'm going to get this right. Okay. So here is a cell membrane. This is your bipolar by drawing it. I'm just going to show you. This is your basic cell. And this is outside the cell. This just goes all the way around. And this is inside. And here you might have sodium ions. Let's just say you have 4. And inside here you have 1. Okay? And so what's going to happen is there's 4 out here, 1 in here, and as long as sodium can pass through the cell membrane, it will come over. Then we'll have 3 over here, 2 in here, then this next one will go in, okay? But then there'll be 3 in here, 2 out here, then another one will go out. It's just a continuous process, and if it can reach where it's the same on both sides, it will, and that's called equilibrium. And you can see that word right here. This is where the concentrations on both sides of the membrane are the same. Okay, let's write that down. This is the condition where the concentrations are the same. Okay, it's called equilibrium. So we have this diffusion and osmosis. And you look at this last term here, it says semi-permeable. And that means the membrane is selective. It allows some things to pass through, other things not to pass through. Depends on what it is. And because the cell membrane is polar, it repels some things. Some things don't have a charge at all, so it's easier for them to pass through. And some things do have a charge, so it's not so easy. So they instead may use channels or pumps to get them across. But this is your osmosis and diffusion. There's a couple more things I wanted to show you. There's these terms. They have to do with osmosis. So we're going to draw an arrow here. And 3 main terms. You have hypotonic, hypertonic. We're actually going to change this here. Go back. That wasn't good. So we're going to erase this. I'd like to color code this a little bit. So hypotonic. Then we have hypertonic. Then we have purple, isotonic. I'm a little colorblind, so hypotonic is, I'm going to explain it to you by drawing you a diagram. Okay, and these all refer to the condition in the cell. Okay, so you look at this cell, this right here, and the baggie is the cell. So here, this is 100% compared to what it's in, which is 70%. So it has a higher concentration. Think hyper. Like a hyper child has so much energy, they have extra. Okay, so this is hypertonic. Okay, and then this one here, the baggie, the cell, has 70%. The 100% in the cell is more. So it has less. It's kind of like a hypodermic needle. So this cell is hypertonic. Okay. Then we have isotonic. That's the condition where they're the same. Okay. So if we have this water, and we have a baggie in it, and this is 50%. Yep, scoot that over. 50% water, and this is 50 in here. They're isotonic. They're the same. They will do nothing. Nothing will get exchanged because they're the same. Okay, so that's isotonic. All things, though, whether they're hypertonic or hypotonic, most likely will reach this because they're going to exchange water, and if possible, they will reach the same concentration on both sides, inside the cell and outside the cell, and become isotonic. Okay, make sure that you can identify that in any cell. If I give you a diagram, make sure you read your unit in that for that assignment, and if you have any questions, please ask. There's some nice animations to watch as well. Make sure you take the time to do that. And some of these words can be very difficult, like hypotonic, hypertonic. They're very similar. The only difference is the O and the ER. Right here and right here. Everything else is the same. Okay, so hypodermic needle, it's small amounts. Hyper, the hyper child, has so much energy they don't know what to do. There's so overflow of energy. Hyper, there's more water in the cell than out. It's always talking about in the cell for these terms. Okay, so that is your notes on the cell membrane and cell boundaries. Again, fill in your sheet, turn it in, make sure you're taking good notes.